Hello, my name is Jessica Slater, and I'm a postdoc in Carolyn Palmer's lab at McGill. Our project applies insights from the neuroscience of musical rhythm to investigate individual differences in ADHD. It's a collaboration with Rita Juber in psychiatry and Simone Della Bella at Brahms. In addition to the diagnostic symptoms of inattention, impulsivity, and hyperactivity, many individuals with ADHD experience difficulties relating to time. Experimentally, this has been observed in a variety of tasks, such as time estimation and interval discrimination, but also in perceiving the beat of music. These behavioral deficits are perhaps not surprising, given several of the brain systems implicated in ADHD are also known to be important for timing. However, ADHD is highly heterogeneous, both in terms of behavior and underlying brain differences, and this is also true when it comes to these measures of timing. Now, we know that different aspects of timing rely to some extent on different underlying neural pathways. So the individual differences in timing seen within the ADHD population may actually provide clues about distinct underlying causes of ADHD. The goal of our study is to identify subgroups of ADHD based on timing-related behavior and associated neural dynamics. We are focusing on two specific components of timing. The first is beat-based timing. This is what allows us to detect the periodic pulse or beat of music, which is what we naturally would tap our foot or snap our fingers to when we hear a favorite song. Beat-based timing relies on basal ganglia circuitry in the brain, and we tested experimentally by presenting participants with a short musical excerpt and a superimposed metronome. We asked the participants to decide, is the metronome on the beat of the music? The second type of timing is interval-based timing. This functions more like a stopwatch, measuring from start to stop how much time has passed. For short intervals in time, say less than 1 second, this relies on the cerebellar pathways in the brain. We assess this experimentally by presenting the participants with 2 short intervals in time, marked by pairs of beeps. We ask the participant to decide, is the second interval longer or shorter than the first? In our pilot testing with a small sample of control participants, we found these measures to be highly correlated. Our prediction is that in a sample of adults with ADHD, these measures will no longer be correlated. Instead, we expect to see subgroups with specific deficits in either beat or interval timing, or no deficits at all, reflecting the individual differences in underlying neural pathways. Given constraints on human subjects research, the EEG component of the study is currently focused on a systematic review of existing literature. We're addressing the question, can EEG identify subtypes of ADHD? We are also working to implement our behavioral testing online so that data collection can continue. The systematic review process aims to provide an objective snapshot of the current literature. This includes pre-registration of the review protocol, peer review of the search strategy, and downloading search results across multiple databases on the same day. I'm now working with my undergraduate research assistants to review our search results and apply our inclusion and exclusion criteria. The goal is to have the manuscript ready to submit in January. Moving our behavioral tasks online has involved research into the many online testing platforms, with a particular concern for timing precision in data collection, and some lessons learned along the way. For example, some online platforms are relatively new and don't yet support important functions like the ability to present sound. Still, we have made significant progress with task development, we have ethical approval, and we will soon be ready to pilot our online experiment. We hope to begin data collection in September. And if the health situation allows, we will invite participants in the online study who are based here in Montreal to come into the lab for further testing, including the collection of EEG data. I would like to thank Carolyn Palmer and members of my lab, my collaborators and research assistants, and of course my funding sources, including my postdoctoral fellowship from HBHL. Thank you.